Hello students, in today's class we are going to learn about prokaryotic DNA replication. So we will first learn about detailed introduction of the replication and followed by the details of the actual process and its regulation. So initially, three different modes of DNA replication were considered by the different groups of scientists, that is semi-conservative model, conservative model, and dispersive model. According to the semi-conservative model of DNA replication, both the strands of the parental DNA will act as a template during DNA replication so that at the end of the replication, each daughter cell will receive one parental DNA and one newly synthesized DNA strand. According to conservative model of DNA replication, during the replication process, both strands of the parental DNA are somehow conserved, but at the same time, it acts as a template for synthesizing two new strands of DNA of the daughter cell. While according to the dispersive model of DNA replication, the parental DNA shreds at random location and is distributed to both the daughter cells, while the gaps in the DNA are filled up by the daughter DNA. So in this way, the parental DNA and the newly synthesized DNA are present in varying ratios according to dispersive mode of DNA replication. However, later on it was identified based on the experiment conducted by Michelson and Stahl that DNA replication indeed takes place by semi-conservative model. So here is the Michelson and Stahl experiment that was conducted. So for this, cells were grown for several generations in presence of heavy isotope of nitrogen so that all the cells will contain 15N heavy isotope of nitrogen. The cells were then lysed and DNA was isolated and was analyzed by cesium chloride density gradient centrifugation. What was observed was a 15N 15N containing DNA was formed at a containing DNA band was formed at a particular position. The cells were then transferred to a fresh medium containing normal isotope of nitrogen, that is 14N nitrogen. After one cell division, the cells were again harvested and cesium chloride density gradient centrifugation was performed. This time, the DNA band was in a new position that was lighter than the original 15N containing heavy isotope of DNA. So the new position was represented as a 14N 15N hybrid DNA position. The cells were again harvested after another cell division and the result showed presence of two bands. One DNA band was at the hybrid DNA band position, while another DNA band was present at a new position that was even lighter. So this was thought to be 14N nitrogen containing DNA position. So the original 15N containing DNA position was not observed after during even the first cell division cycle. So these experiments suggested that indeed the DNA replication is taking place by semi-conservative mode. Now, the next slide talks about the accuracy of DNA replication process. The process takes place with a very high rate of fidelity or accuracy. During replication, the E. coli cell makes about one mistake every 10 to the power 9 to 10 to the power 10 nucleotides added. So if you compare this number with the size of the genome, which is about 4.6 into 10 to the power 6 base pairs, so which means the accuracy rate is about 1000 to 10,000 times the size of the genome, which means that every 1000 to 10,000 replication cycles, one mistake is done by the E. coli DNA replication process. So how does cell ensures this high rate of accuracy? So during polymerization, the discrimination between the correct and the incorrect nucleotides relies on several factors. First of all, the hydrogen bond. This ensures that the correct base pairing takes place between the complementary bases. That is, an adenine will always pair with a thymine because of complementary base pair. Then the geometry of the base pairs. So basically, in a base pair, we have a purine and a pyrimidine, which is one of them is a single ring structure, other one is a double ring structure. So they have a typical geometry that is accommodated or fits well into the active site of the enzyme. So sometimes an incorrect nucleotide may hydrogen bond with the base, but it should also fit into the active site of the enzyme. So this forms the first level of measure by which the incorrect base is rejected even before formation of the phosphodiester bond. But still, the polymerase may insert one wrong or incorrect nucleotide for every 10 to the power 4 to 10 to the power 5 correct nucleotides that are added. And the reason for this is the tautomeric forms of the bases, because of which sometimes a wrong base will still fit or adjust into the active site and form bond with the corresponding base. So then we have another activity of the enzyme DNA polymerase, which is known as proofreading activity. This activity is basically 3' to 5' exonuclease activity, which is an activity that runs in opposite direction of polymerization. Polymerization takes place in 5' to 3' direction. So if any incorrect or incorrect base is being added and the phosphodiester bond is also formed, then still the polymerase can later identify that uh, this is an incorrect base added. So with the help of its proofreading activity, it will remove the incorrect base and then it will proceed with the polymerization again. So proofreading activity will improve the accuracy by about 100 to 1000 fold. So if we combine base selection and proofreading, the combined accuracy is about one error per 10 to the power 6 to 10 to the power 8 bases. If we compare this number with the actual efficiency, which is about 10 to the power 9 to 10 to the power 10 bases will have one mistake, we can still find that there is a difference of about 1000 to 10,000 times. So this rest of the accuracy is accounted by the post-replicative repair mechanism, which will repair any mistakes that have escaped all these methods, improving the efficiency, and which will account for the actual efficiency, which is one mistake in every 10 to the power 9 to 10 to the power 10 base pairs that are polymerized. Then this slide discusses about the comparative properties of the three types of polymerases in E. coli: DNA polymerase 1, 2, and 3, which are encoded by the genes polymerase A, polymerase B, and polymerase C, respectively. They have a molecular weight of about 1,03,000, 88,000, and 7,91,000 Daltons, respectively. The proofreading activity is present in all the three enzymes, while the 5' to 3' exonuclease activity is only present in DNA polymerase 1, which is actually an enzyme that is involved in the DNA repair process. If we compare the polymerization rate, that is the number of nucleotides added per second by the three enzymes, DNA polymerase 1 can add about 16 to 20 nucleotides per second. Polymerase 2 can do 40 nucleotides per second. while polymerase 3 has the highest polymerization rate with up to 1000 nucleotides per second. Then comparing the processivity is basically the nucleotides added before the polymerase falls or dissociates. So during polymerization, after adding a fixed number of bases, the polymerase falls off from the DNA, and again it has to sit on the DNA and continue with the replication process. So processivity is basically the number of nucleotides that the enzyme will add before it dissociates from the DNA. So here also we can see that the polymerase 3 has the highest processivity, about more than 5 lakh nucleotides it can process at a time. So based on these properties, it could be understood that the DNA polymerase 3 is the main replication enzyme, and the other two polymerases are actually now known to be involved in the DNA repair process, or towards the end of the replication process, they may assist. But polymerase 3 is the main replication enzyme. Now, the next slide talks about the origin of replication in E. coli. So, origin of replication is the sequence where the process of replication will begin. It is about 245 base pairs in length, and it consists of the four 9 base pair repeats and the three adjacent 13 base pair sequences. The 13 base pair sequences are AT-rich regions, and they also contain a conserved GATC residue. During replication initiation, about 20 DNA A protein molecules, each activated by binding ATP, will bind the four 9 base pair sequences, during which the DNA basically wraps a group of about 20 activated DNA A proteins. This then results in sequential denaturation of the adjacent 13 base pair AT-rich sequence. As the DNA denatures at the 13 base pair sequences, the next protein that enters is the DNA B protein. So DNA C protein here basically helps in recruitment of DNA B protein, while DNA B protein is a helicase, and it is basically a hexamer made up of 6 subunits. So two such helicase complexes will be polymerized along each strand of DNA, initiating formation of a replication complex. So overall, two replication complexes are, or two replication forks are generated here. And now the DNA is ready for next step, that is priming. So at intervals during leading strand synthesis or formation of Okazaki fragments, the enzyme DNA primase will work in close association with helicase and will synthesize a stretch of RNA primer. The primer is then extended by the DNA polymerase, and it will extend the primer till the previously formed Okazaki fragment is reached. Then again a new primer will be synthesized to begin the process, and in this way it is repeated. So the replication process in prokaryotes is a highly regulated process, and only the initiation step of replication is basically regulated. Regulation of replication also involves activity of another protein that is known as DNA A protein. So the timing of the replication initiation is affected by DNA methylation and interaction of the origin sequences with the bacterial plasma membrane. The ORIC or origin of replication is methylated by the DAM methylase activity in which the adenine residue is specifically methylated within the GATC sequence. There are about 11 such DATC sequences present within the 245 base pair region of origin, and as such, in the rest of the chromosome, about one GATC sequence is present in about 256 base pairs. So immediately after replication process, your DNA strand gets separated and you will have synthesis of the daughter DNA. So the original parental DNA, both the strands of DNA are basically methylated and they are known as completely methylated DNA. But when the daughter DNA is synthesized, you will have the parental DNA is methylated, but the daughter DNA is not methylated. And the hemimethylated form of the origin sequences are sequestered by the plasma membrane for some period of time. After a time, the origin is released from the plasma membrane, and before that, the other strand of DNA must be completely methylated, and this is done by the activity of DAM methylase. So after completely methylated form of DNA is formed, the DNA will be released from the plasma membrane and then it can enter into the respective daughter cell. The regulation of initiation process also involves the activity of DNA A protein. So the active form of the protein is required to initiate the replication process, which is by interaction with ATP molecules. And after initiation, DNA A protein will become inactive and needs to be reactivated again, which requires a time gap of about 20 to 40 minutes to become active. So unless the DNA A protein is active, replication cannot begin again, and it takes time around 20 to 40 minutes depending on the bacteria, which is in parallel with the cell division time. So therefore, only after cell division, another replication cycle can begin. So in this way, the two mechanisms, that is DNA methylation and the activity of the DNA A protein, ensures that the DNA is replicating only once during each cell division cycle. So here we have a summary of different proteins that are required in initiation of DNA replication. So DNA A protein recognizes the origin sequences. DNA B protein is a helicase that is involved in unwinding of the DNA. DNA C protein helps in recruitment of DNA B protein. HU are histone-like proteins that stimulates initiation. Primase will synthesize the RNA primers. SSB proteins will bind single-stranded DNA, stabilizing them. DNA gyrase functions to remove the torsional strain that is generated by DNA unwinding. And DAM methylase will methylate the DNA at the origin sequences at the GATC residue. Now, elongation phase of replication includes two distinct but related operations, that is leading strand synthesis and lagging strand synthesis. Several enzymes at the replication fork are important to the synthesis of both the strands. Parental DNA is first unwound with the help of DNA helicase, and the torsional stress generated is relieved by enzyme topoisomerase. Each separated strand is stabilized by SSB protein, which is single-stranded DNA binding protein. Now, before we proceed into the details of elongation, we must first look at the different subunits of enzyme DNA polymerase 3 and their role before proceeding. So DNA polymerase works in close association with the helicase enzyme which carries out unwinding of the DNA. DNA polymerase 3 is made up of two core polymerases. Each core polymerase is made up of one subunit each of alpha, epsilon, and theta subunit. Then we have a clamp loading complex which is made up of two tau subunits, one gamma subunit, one delta, and one delta prime subunit. The clamp is basically a clip. So this is the beta clamp, and it works in close association with the core subunit in order to improve the processability of DNA polymerase. So they are basically a clip-like structure which helps to hold the DNA so that the core polymerase can carry out the extension with optimal processing. So DNA replication basically will take place in 5' to 3' direction. Now, in case of leading strand synthesis, the replication is taking place in the same direction as the unwinding of the DNA by the enzyme DNA helicase. So because of this, the leading strand synthesis will take place continuously. While lagging strand synthesis is taking place in the opposite direction, it is again in 5' to 3' direction, but it is the opposite direction of the unwinding of DNA by the enzyme DNA helicase, and hence lagging strand synthesis will take place in fragments. So, primase enzyme will enter in the next stage. So, primase enzyme occasionally associates with the helicase, and they together form a functional complex which is known as a primosome. The primase will synthesize a new primer and then dissociate during lagging strand synthesis. Now, the beta clamp loading complex loads a new beta clamp onto the newly synthesized primer. Simultaneously, the core polymerase continues with the extension of the previously formed primer till it reaches the beginning of the previously synthesized Okazaki fragment. When the synthesis of Okazaki fragment is completed by the core polymerase, the enzyme will take a halt and it will be dissociated from its beta clamp, and the new beta clamp that is associated with the primer will then be transferred onto the core polymerase. And now the core polymerase will again continue with the extension process. So the entire complex responsible for coordinated synthesis and replication at the replication fork is known as replisome. So the same points are mentioned in this slide for your reference. Coming to the final steps in the synthesis of lagging strand. So once the Okazaki fragments have been completed, its RNA primer needs to be removed and replaced with DNA. So this is done by the activity of enzyme DNA polymerase 1 and by a process which is known as Nick translation. And at the end, only phosphodiester bond formation will be remaining, which is known as a Nick. And this Nick or formation of the final phosphodiester bond is done by enzyme DNA ligase. Then coming to the last stage, that is termination. So eventually the two replication forks that move in opposite direction will meet at the terminus sequences. So the terminus sequences are arranged in the chromosome like a trap, and so that the replication fork can enter the trap but cannot leave. These terminus sequences are present on towards the opposite end of the origin, and these sequences function as binding site for a protein which is known as the TUS protein, which stands for terminus utilization substance. The TUS-TER complex arrests the replication fork from one direction only, and only one replication complex functions. Only one TUS-TER complex functions during the termination. The process ends when the second replication forks meet the arrested replication fork, completing the replication process. Coming to the last stage, that is chromosome separation. So at the end of the replication process, the two strands of a DNA are entangled with each other because of the activity of helicase and followed by the activity of topoisomerase that again and again cleaves the parental strands and relieves the torsional stress. So as a result, at the end of the replication process, the two daughter strands that are synthesized or the two daughter DNA chromosomes that are synthesized are intertwined or entangled with each other, and they are known as catenated forms of DNA. So here enzyme DNA topoisomerase 4 will function, which will help in decatenation or separation of the chromosomes. So they will transiently break the phosphodiester bond of both the strands of one of the daughter chromosomes, allowing the other chromosome to pass through the break, and then the phosphodiester bond is reformed. So in this way, the daughter chromosomes are separated from each other, and then they can enter into their respective daughter cells. So that's it about prokaryotic DNA replication process. Kindly like, share, and subscribe if you like the content. And thank you.